Okay. All right. Hi, everyone. This is actually part 2 of this week's lecture. I did part 1 a couple nights ago. So anyhow, with this presentation, I will be talking about some additional structures. On the first presentation, I ended up talking about the prokaryotic cells, and specifically more about the ribosomes and about the DNA, which is in a nucleoid. And I also talked about the cell membrane and spent a lot of time talking about the cell walls and the different structures of cell wall and the organization of peptidoglycan in those different types of cell walls, or the lack of. So anyhow, tonight or today, I'm going to be talking about some additional structures on the outside of the bacterial cell and a few additional structures that some bacteria have on the inside, or the cytoplasm of their cells. So the first thing here, starting with slide 16, is going to be the glycocalyx. Now this is like a sugar coating. It surrounds the bacterial cell outside of the cell wall. It's made of polysaccharides, proteins, or both. And there are 2 types of glycocalyx. One is the slime layer. The slime layer is almost like it sounds slimy. It's very loosely organized. It may include some lipids. It's easily washed away or off of the bacterial cell. And some of the functions, or the main function of the slime layer, is to protect the bacteria from drying, and also to collect or I guess accumulate nutrients for the bacteria. Now, a second type of example of glycocalyx is going to be your capsule. Now, not all bacteria have a capsule, but if they do, this is a very thick layer. It's attached tightly to the bacterial cell. It cannot be washed off, and the function of the capsule is really to protect the cell from the host immune defenses, in particular phagocytic cells that may see it as harmful and will want to attach and eat the bacterial cell. The capsule will protect it in that it actually will cover or hide any type of protein that the defense cell will want to attach to or see as foreign. So now, in addition to the glycocalyx, also some bacterial cells and archaeal cells will have a single layer of additional protein and glycoprotein. This is your S layer. And I think I pointed it out the other night, or for the previous presentation, that some, the gram positives do have an S layer outside of their cell wall. Archaea have an S layer too, but it's a little different in that its composition is different, but it is the cell wall. So, okay, I wanted to say that the bacteria that produce the slime layer will also build biofilms. This slime layer helps in building a community of bacteria known as a biofilm. And what this is biofilms naturally occur where there's a water source, and you'll see them in the environment naturally, but they're finding out now that these biofilms are present in many chronic infections. And there will be a video later on about biofilms and the detailed development of them. But just know that these bacteria do secrete a slime layer in the initial stages of biofilm formation. Okay, some of the appendages or extensions that appear or extend from the bacterial cell are filamentous, which means they're tubes. And one of them is a pili, and this is made of a protein called pillin. Okay, there are two types of pili. One is fimbriae, and these type of pili have adhesins, or like a gluey factor that allows them to stick to surfaces. The other type of pili is called a conjugation pili, and this pili is used to connect one cell to another in order to transfer genetic material that is found in a plasmid. Okay? They're both used for attachment or connection, but for different reasons. Okay? Another type of appendage, it's not a pili, is a stalk. And it should read stalks secrete adhesion factors. And these stalks are actually found on some bacterial cells, and they are extensions of the cytoplasm. Okay, so they aren't protein tubes like you would find in pili, but they are the cytoplasm kind of forming the tubes from the bacterial cell. And again, though, these are used for sticking to surfaces. All right, there is another type of extension that a bacterial cell may have, and this is found on bacterial cells that are motile, that move. And these are called flagella. These are made of flagellin. Okay? Again, my face is blocking that, but if you have the PowerPoint slides in the lecture material, you'll see that says flagellin. And how those work is that the flagella is anchored into the cell wall and cell membrane. And what's anchored there is a type of motor that the flagella has. And then there is the tube, the flagella tubes that make up the flagellum, extend from the body of the bacteria. Now, how it works, to put it simply, is that it's kind of like a motor, outboard motor on a boat. And you've got the engine part, so that would be the part that's embedded in the bacterial cell. And then you have the propellers. And the propellers would be that long tail, the flagellum. And it's the one that rotates, okay? And it can go either clockwise or counterclockwise. If it goes counterclockwise, the bacteria will move in a forward or straight line. If it goes clockwise, the rotation will switch to clockwise. The bacterium will flip. And what purpose does that serve? Well, if it's going towards something that sends out signals as being harmful, or harmful or not beneficial for the bacterium, it will switch into clockwise, flip, and change direction, and then propel counterclockwise. So it's known as chemotaxis or taxis. Okay. Now, we do have, usually the flagellum extends from the bacterial cells, but there's one exception. And that exception is with the shape, the spirochete, the bacteria that's shaped like a spirochete. They do not have a flagellum if they're motile. They do not have an exterior or external flagellum. Instead, their flagellum wraps around the body of the bacterial cell, and it's covered by an outer sheath. And what happens is, when the body of the bacterial cell constricts, it will exert a force on that endoflagella, or another term is axial filament. And that pressure, the result of the constriction of the bacterial cell, will cause the bacterial cell to move. Not only move, but rotate. So the whole bacterial cell will rotate like a corkscrew motion as it moves. So that is the exception for the external or exterior flagella. The spirochete has its own special little type of flagella, okay? That's important to know. Okay, so now we're talking about the external flagella arrangements. They do come in arrangements. And monotrichous. So this is a bacterial cell where the flagellum will be on one end, or polar. Polar is another word for end of bacteria, the ends of the bacteria, or end of a bacteria. So the flagella arrangements in your monotrichous, one is one flagellum, and it's on one end of the bacteria. In your amphitrichous, you've got one flagellum on both ends, okay, or both poles of the bacteria. I try to think of the word amphibian. If you think of amphibian, it's late at night, people, sorry. That an example of that would be a frog. So it lives part of its life in water and also on land, both. And that's what amphitrichous means. It means that you've got one flagellum on both ends of the bacterial cell. Lophotrichous, I have no neat way for you to remember that. Just know that it's two or more flagella on either one pole, one end, or the other pole, other end of the bacterial cell. Finally, we have peritrichous. Peritrichous is all around. If you think of perimeter, those flagella are all around the bacterial cell. And if you look at the picture below it, you'll see those bacterial cells, and it looks like they've had a bad hair day. Okay, all those little crinkly extensions are flagella. Now, if you have time, you can look at the little video down there. It'll show you some bacteria moving, motile by flagella, and one little guy stuck to a cover slip, so he doesn't really go anywhere. He just kind of spins. So, if you want something to look at, it's very short and kind of cute. Okay, now I'm kind of working into the interior, the cytoplasm of bacterial cells, and these are additional structures, so they aren't found on every bacteria or in every bacteria cell. But we do have some inclusion bodies, and I'm going to be talking about some different types of inclusion bodies in the next few slides. The one main guy here is your metachromatic granule. Another name for it is volatin. And these will show up usually in your stained smears because when bacteria is unstained, you really can't see the bacteria or any of its structures. So in a stained smear, you'll be able to notice these, and their function is to store certain nutrients that the bacteria may use, such as that first one. You don't need to know the name of it, but it is a lipid. Also, they may store stuff like phosphates, sulfur, or sugar, polysaccharides. An example of metachromatic granules is your Corynebacterium diphtheriae. So below are 3 different stains using that bacteria and showing the metachromatic granules. In the nicer stain, you'll see that the green rod-shaped part is your bacterium. The little, I don't know if they're purple or brown dots, kind of look like matchsticks. Those little purple or brown dots are your metachromatic granules, okay? The methylene blue, it's not quite as nice as a nicer stain, but if you look at it, the lighter shaped rods here are going to be your bacterium. And that dark solid blue, they do stain solid, is going to be your granule, metachromatic granule. And finally, the gram stain, which you're going to be doing a lot of this semester. Again, it's not quite as nice as the first or even second picture, but in the gram stain, you've got the lighter colored bacteria with the dark solid purple dot at the end, and that is your granule. So in a lot of different types of stains, you will be able to see these metachromatic granules. So the next slide, I'm going to be showing other examples of inclusion bodies. And some of these inclusion bodies are have some pretty neat functions. So let's look at that PHB granule, and this is in a bacterium of Pseudomonas. That's going to be on the left side of the slide there, the upper left side. And those are actually rich organic stuff. So it's, as I said, these granules are storage. So I kind of like to think of them as freezers or pantries. And usually they will form when there's a lot of nutrients available. It's kind of like going to, you have a grocery store. Let's say Hannaford's is having a great sale on ribeye steak. Buy one, get two free. Of course, you're going to pick up a bunch of them and stick them in the freezer for future use. This is pretty much the reason these inclusion bodies do form. So these nutrients are available, and they kind of hoard them. So like I said, in this first one, this is some organic stuff. This is food that the bacteria will use. So it will form these granules that will contain that rich organic stuff for later use. Okay. All right. In B, we've got a bacilli, and it has a crystal, BT crystal, just a short name for it, and it's where the arrow is. It's kind of a light colored gray. And this bacillus forms these crystals. They contain protein, and what happens is, actually they do not cause us harm, but they do kill insects. They harm insects, and sometimes this bacillus is used as a type of safe insecticide, and they will apply this bacterium around the base of a house, or in an area where insects are a problem, maybe carpenter ants, termites. And the bacteria, once it enters that insect, it kind of cuts the insect's gut like glass. So it's a safer form, I guess, of insecticide instead of using toxic chemicals. The third one is carboxysome. And this is usually found, it's right here, in this picture here, usually found in photosynthetic bacteria. And what it does is it contains enzymes for carbon fixation in a stage of photosynthesis. Okay, so you do see it in bacteria that photosynthesize. Finally, the sulfur globules, indeed, they look like pretty little shiny crystals there. These are all sulfur. Sulfur's in these granules here. Okay, and they almost look pure white if you look at them. With a compound light microscope. They're beautiful. And again, this is sulfur, and these are accumulated and stored in the bacteria because these bacteria use sulfur as their food. They're found at the bottom of oceans where there isn't a lot of there aren't a lot of nutrients down there. So sulfur is usually available, and these bacteria can grow there. Okay, now these are a special kind of inclusion bodies, and they're pretty neat. They're magnetosomes. And these are found in some bacteria that have magnetic properties. So what happens is, basically, their food is iron sulfide. And you find that a lot of times at the bottom of standing water sources. So their food is iron sulfide, and then once they use the iron sulfide, magnetite is produced, or is a byproduct, and magnetite is stored as crystals in a chain that's called a magnetosome. So if you see there, sometimes there's 15, 20, 25 of these little granules linked together into a chain. You'll see that on the top bacteria there. Those are magnetosomes. And again, they have a magnetic property. They act almost as a compass, so that they are drawn to the Earth's magnetic field. They sink to the bottom of water sources, and two reasons. That's where it's pretty clever that they've evolved this way, but that's where their food source is. And not only that, but there's very little oxygen there, and they survive. They love low oxygen conditions. So these are your magnetosomes, and they're fairly common. You can find them in a lot of areas, even around here, or a lot of water sources. Now this is another inclusion, endospores. Now not all bacteria produce endospores. And I'm going to tell you the shape of the bacteria that you will find them in are your bacilli, okay? You won't find them in causae, you won't find them in spirochetes, in vibrios. They are found in bacilli, but not all bacilli produce endospores. But those that do produce endospores are bacilli, okay? Now, why do they form? They actually form because there are harsh environmental conditions. So it's a drought, there's not a lot of water, there aren't a lot of nutrients, it's very hot, very cold. And as a result, these specific bacteria that can form endospores will. Now, what are they? They are really a type of bacterial reproduction. It's at the end of bacterial endospore formation, you will end up with a hard, dormant, inactive, non-metabolic bacterial cell. And what it does is it will persist in its environment, whether it's soil, whether it's rocks, mountains, whatever. It will survive there until the conditions become favorable, and then what happens? It turns into a viable, living bacterial cell. And I know I was talking to you about sterilization in our autoclaves, and I said this is the only thing that will kill endospores, because when this endospore is in endospore formation, it's very, very hard to kill. It's very resistant to heat, to chemicals, to cold. But sterilization with an autoclave will be able to kill it, okay? That's the only thing. These guys can last for years and years and years. In fact, I think there was a mummy, a 3,000 1,000,000-year-old mummy that they found endospores on. Okay? I'm getting towards the end here, guys. So anyhow, I'm going to talk a little bit, or show you a picture here, a little bit of the characteristics of eukaryotic cells. And I did mention before that they are larger, much larger sometimes, than your bacterial cells. They do have a nucleus, and inside that nucleus is your nucleolus, which that is certainly lacking in bacterial cells, along with the nucleus. The eukaryotic cell will have an internal cytoskeleton. So these are fibers that run throughout the cytoplasm, and they help to keep the organelles within the cell in place, but they also help keep the cell structure of the eukaryotic cell. You have an endomembrane system, which would be examples of that would be your endoplasmic reticulum, basically, and the Golgi apparatus. You will have cell organelles, mitochondria, and chloroplasts, which I will talk about in a minute. Very, very interesting about them. Golgi apparatus, and you may have some intracytoplasmic bodies, unlike the granules in prokaryotes. In this case, those inclusion bodies will be vacuoles and lysosomes. Anyhow, so let me talk a little bit about the mitochondria and the chloroplasts, and this is going to be ending this week's presentation when I'm done here. Okay, there's some speculation that the mitochondria and the chloroplast, it's more than speculation, folks, were at one time bacteria, okay? And it's more than speculation. What conclusion they've come to is that it was bacterial in origin because with all this wonderful technology and research, it's been determined that mitochondria, along with chloroplasts, do have a cell membrane. They have their own DNA. They've got their own ribosomes and enzymes. That sounds like, man, that is certainly a prokaryotic cell. It's got the must-haves, okay? The ribosomes for the mitochondria are the same structure as the ribosomes of prokaryotic cells. Not the ADS, which is the size and the structure of a eukaryotic ribosome. And then what they did is they sequenced some of the mitochondria's genome. And what it showed is they are closely related to a group of bacteria known as alpha proteobacteria. So there's a lot of evidence, strong evidence, pointing into the direction that mitochondria were originally bacteria, prokaryotes. Now, how does that fit into the big scheme of things? And this article kind of explains it. It actually explains two theories, okay? Two theories. Generally, the theory is called the endosymbiotic theory. And what that is that at some point that mitochondria came about because one of two reasons, or one of two ways. A bacterial cell was acquired by a primitive eukaryotic cell, and this bacterial cell was oxygen loving, so it would use the oxygen and make energy. That was acquired by a eukaryotic cell, and it was a symbiotic relationship. They both pretty much benefited because the oxygen loving bacterial cell, which now was part of the eukaryotic cell, had protection. And the eukaryotic cell was reaping the benefits of extra energy that was released by the prokaryotic cell. An endosymbiotic relationship. Later on in evolution, there was a second symbiosis in which you had that the chloroplast, or a photosynthetic bacteria, was engulfed by the eukaryotic cell, and a chloroplast was born. The chloroplast. So that is one theory, but it's an endosymbiotic theory. You had symbiosis going on. It was beneficial for both. Mutualism, I would say. That is one theory, the endosymbiotic theory. Another one that this article raises is the idea that it was actually a bacterial cell and an archaea cell. Again, that's another prokaryote that converged. And they developed or evolved into the eukaryotic cell. So that's just kind of a new twist on the endosymbiotic theory. But it just seems it's just great that to realize the whole scheme of evolution. We got here, our cells got here because of those cells. Okay. So anyhow, that's going to be it for now, or for tonight. And I do post a little review at the end of these PowerPoints, but I just want to tell you that's just for you to keep up weekly with the material. I will have in fact, I did post a review for the exam that's coming up next Friday. So you can print that off for your viewing pleasure. Again, if you have any questions, you can either email, sign on to one of my Zoom meetings, or even ask me in class or in lab, okay? All right, you all have a good week, and I'll be seeing you soon. Bye.